All right, so a couple of things. First thing, I did finally meet with the Wiley Plus person and got lots of help yesterday. So I should be like all set now. Of course, some of the things I found out the hard way. Like letting you guys convince me to put a link that they told me not to put in my course to all the assignments. That's why your grades aren't showing up. We're not showing up. If you use that assignments link that listed all the assignments, if you went there to do your assignment, that's why your grade is not showing up in Canvas. So they're going to do a grade sync for me, so hopefully that will get fixed. So that will not be turned back on. You will have to go to the links that I put in the modules. And honestly, y'all, it really doesn't matter whether it's a Wiley assignment or not. But if you really want me to, right, in the name of the assignment, like off to the side, you see what I did yesterday? I could write Wiley Plus. Okay, I learned how to do that. So I also thought in the other program that I used to use, if I gave you guys unlimited attempts, that like you literally could take the whole assignment over again. Wiley Plus does not do that. Once you get a question right, it doesn't give it to you again, right? Anyone done the assignments more than once? Okay, right? So it just gives you another attempt on the ones you got wrong, right? And it just shows you, I guess, the ones you got right. Okay, and so therefore you know what the correct answer is? Yes? Okay, because you got it right. Okay, so I think I'm going to leave it unlimited attempts. So one of the things that you guys wanted was to get hints and stuff, like links to the textbook or given the answers after a certain point. So I think what I did on the last one was that after you take it one time, the next time you go to take it, any of the ones you're having trouble with, you could click the link and see a link to where hopefully in the textbook it would help you. Does that make sense? And then I set it up so that once you finish that attempt, if you still got it wrong, when you open up the third attempt, did he explain this to me that way? You could click if you don't know what the answer is and it will tell you. Now each one of these like choices, y'all, it's a nightmare for an instructor. Like I could take points off and there's so many choices. Yeah, okay. It's insane. I don't want to do that, and I won't probably ever do that. I will never like take points off for you guys, right? I want to make this tool usable for you and for me and not make anybody's life a nightmare, right? The reason for these assignments, and I said this last time, is for you to self-quiz yourself, right, before the test. So I want you, the very first time you take it, to not have any notes or books or anything open and see what you actually know, right? So also study the material, right, as if you were going to take a quiz or a test before doing these assignments. Still with me? Because guess what? You can do it again, right? And the next time you can even get hints, right?
So that grade, the quiz grades don't really, if we do it again and again, it doesn't really affect anything, right?
So if you keep doing it, you should eventually at some point get them all right? Unless, you know, I don't know how to say, I don't know how to say it. All right, so I expect you guys to max out your points on all of these, right? All you have to do is do it. Does that make sense? I don't want to take points away from you guys for like doing it multiple times or anything like that. The only thing I'd be somewhat tempted to do is take off points after the due date. But again, I don't want to do that because, you know, life happens. And then some student's going to come to me, you know, and I have a lot of students, so it's not just one or two, it's like 5 or 6 come to me like, but this happened and I missed the due date. Can you please not take those points off? And I feel like I don't really need to do that either because, quite honestly, if you don't do it by the due date, or at least before that exam, you're only hurting yourself, right? Because then you go into that exam unprepared. And guess what? You're going to do bad on the test. Some of you guys already know this, right? It's already happened. So I don't need to basically penalize you twice, right? You're adults. You know what you need to do, right? When I used to set up these assignments, I used to actually put the deadline at the end of the semester. But students wouldn't do the assignments because the deadline was at the end of the semester. I'm like, no, no, no. That's because I want you to be able to do them again for the final, which in this system, I really can't do, right? So I set my deadline because that gives you guys a guideline, right, on when you should get this done by. But if you don't get it done by the end of the semester, it's whatever it is, it's a 0, period. Right? But if you wait that long, you're only hurting yourself. Does that make sense to you guys? So I'm going to keep putting deadlines, but they're not hard deadlines. You're not going to get penalized if you answer it an hour or a day late. The important thing is definitely get it done before you take a test on it, right? Because if you don't do it before the test, you're not going to know. And then you're going to take a test and you're going to think you know it and find out, oh crap, I don't really know this. Does that make sense to you guys? Okay, so that's what I figured out and what my plan is as it relates to future Wiley assignments. So they'll always be unlimited. I'll put a due date to help guide you, but you can do it after the due date, right? But it's in your best interest to get it done by the due dates, right? Which right now, I'm just going to set them the day of the exam. But again, do them before then, right? I'm not going to penalize you for doing it late. And again, once you get them all right, there's no point in doing it again because it's all done. Does that make sense? Okay, so then the other question I was asked, Vanessa asked me this morning, was can we take the test again for practice? And I have done this in the past, where towards the end of the semester, I made the test available in a practice setting. But I think actually, I'm glad she asked this question. I think it'd be more beneficial that why this stuff is kind of fresh in your mind, especially for some of you guys are looking at your test results and going, I'm not exactly sure why I got this wrong, right? So I'm going to make it available as a practice, which means it doesn't show up in the grade book, 0 points, where you can basically take the test again, right? And so you could figure out where you went wrong, or maybe study again and see if your change study methods are helping you improve. But there's a warning on this. Remember that these questions are not the questions on the final, right? They're going to change. It's the same objectives. Whatever objectives are listed in the test is what has a chance of being on the final, a 50-50 chance of being on the final. Does that make sense to everybody? So I'll do that for both my face-to-face and my online class. And so the other assignment for face-to-face class that you guys have coming up was the agreement. And I didn't realize it was already on. So if you guys have done it, and of course it says what type of assignments in it. Connect, right? Because that's what we used last semester. And I hadn't changed the assignment before a few of you guys did it, so I don't know even if I can change it, but I'm going to try and change it. But know that you guys that already took it, what I just talked about as it relates to Wiley, that's the way it's going to be, right? So I will fix that after class today and post an announcement. For online students, they had a different agreement, right? They got a whole crazy testing schedule, so. Theirs is about when they're going to take the test and where they're going to take the test. For you guys, it's, assignments and reminding you, yes, that test that you took, it's not being dropped. No tests are dropped in this class. Right? Any other questions as it relates to stuff outside of lecture? Okay. So you'll see that I started putting the stuff in Module 2, and I'll build on that probably a lot this weekend. So if you have announcements set up, you probably get a whole lot of notifications from me this weekend, because I'll probably try and build out the whole module this weekend. Lockdown browser. That's what you have to review your test, to take the practice test I'm going to make available to you, and to do the agreement assignment, because part of that agreement assignment is so that I know that you have access somewhere, somehow, whether going to the computer lab or at home, to the lockdown browser so you can review your tests. Right. So you need to call tech support because that's their job. And otherwise, go to the computer lab upstairs on the second floor here, or the Hibernian Center where you guys took your test, or my office. Right? So if you can't get it to work at home, that's something you need to call tech support to help you with if you want to achieve that goal, but there are other places you can go on campus. Yeah. I think some people have had it work on a Chromebook. I think it's a, so wait a minute, Chrome is like a PC thing. Does it have apps? There is a Respondus app. I don't know if they have it for, I know they have it for Apple. So any Apple products, there is a Respondus app. But I'm not sure about Google Chrome. I know I had some students using it's a tablet kind of thing, huh? Or, yeah. Oh, right. There's no programs on it, but there's apps, right? So there might be an app. Might. Yeah. Let me know. Because I know they're becoming more popular. I know at my son's school they're suggesting we buy that for him. Cheap? What do you consider cheap? It's like $300-400, right? Where? Oh, yeah, you didn't buy new. Okay. New, they're like $300-400. Okay. Good for you. Bargain shopper. All right, okay, so that wasn't too bad, like, what, 15 minutes? Okay. All right, I'm not going to do my disclaimer. Okay, so last time we were starting out stuff in Chapter 7. We talked about the Griffin experiment, right? How he figured out, right, there was something coming from one set of bacteria being picked up by the other. Turns out it's DNA, right? But it wasn't really confirmed until 2 other experiments were done. So one of them being done by these 3 scientists, Avery, McLeod, and McCarthy. And they attempted to determine which molecule it was. Was it DNA, was it RNA, or was it protein that caused the cells to transform? Right? We know today that it's DNA. But we know this because of scientists like these guys that came before us and worked this out. So they used mixtures of the smooth strain, which remember is the one that's able to produce the capsule and therefore is pathogenic. So they only mixed DNA left over with the R cells and that turned them into S cells, right? Indicating that DNA was the molecule responsible. But how did they actually do this? They use proteases and RNases and DNases. These are enzymes that digest each one of those molecules. So they digested away any proteins that were in that extract. And they found transformation still happened. They digested away all the RNA and transformation still happened. But when they digested the DNA, no transformation, right? So that confirmed, okay, it is just the DNA that is going to allow them to transform, allow to have this new ability they didn't have before. Of course, some scientists, although this is pretty good experiment, right? We're still not convinced. So later on, Hershey and Chase built upon this. And they used radioactive labeling. So they labeled proteins in DNA. And part of the reason for this is that, remember, DNA wraps around proteins, especially in eukaryotic cells. Anyone remember the name of those proteins? Histones, right? And same thing in archaea, right? They have histone-like proteins. But bacteria, too, have proteins that help them condense their DNA, that their DNA wraps around. So some scientists weren't convinced that it was just a pure DNA extract. So bacteriophage that we're going to learn about in our next topic when we talk about viruses, these are viruses that infect bacteria. And in fact, it's only the DNA that goes into the cell, not the protein code. And so because of that, they were able to tease out yet again, it was the DNA that actually causes the transformation. Does anybody know what element, right? So things like on the periodic table, like carbon and nitrogen and oxygen. You know what element is found just in proteins that they radioactively labeled? Sulfur, right? And what did they label in the DNA with a radioactive label? What element? The letter is P, but you say, what is P? Phosphorus. Phosphorus. Yeah, phosphorus. So again, they were able to, with the radioactive labels on those elements that are only found in those two different molecules. So this is a good preview to what we're going to be talking about later too when we talk about what elements make up these molecules, right? I'll put that picture in your brain. So the labeled phage DNA is the only thing that went into the bacteria. The proteins were left on the outside. So it's a really cool experiment, right, where they separated out and you could see, right, the DNA was inside the cells where the protein was outside the cells. So now we get into the structure. So this was before even the structure was known, right, of DNA. So everybody knows about Watson and Crick. Most people know nowadays that they also worked with another scientist whose lab mate who did most of the work, Rosalind Franklin, did not get credit at the time. She actually was dead when they got the Nobel Prize. She had died of cancer. But posthumously now, like, we give her credit for her contributions that led to the discovery of the structure of DNA. And the structure is really important to its functionality, which is why some components of this I'm going to review with you guys. And again, because biology may have been a while ago for some of you, or you didn't cover it so well. So it's a 5-carbon sugar, and that particular sugar is not a complete 5-carbon ribose. It's missing some oxygen, right? So that's why it's called deoxyribose. And that's the D part of DNA, right? Deoxyribose. The N is nucleic acid. The N is nucleic, A is acid. So the other part of the molecule, the phosphate group that binds to the sugar, right, that hooks the different nucleotides together, and then off of the sugar on the other end you have the nitrogenous base. And they attach to a particular numbered carbon in that sugar. So when we look at it, and I hate that the pictures are so small. I already cut out one picture earlier and put it on a slide all by itself. What'd I do? There we go. Now we can see it much better. So we know it's double-stranded, and part of how they figured out, you know, what letters went with what was, of course, important to its structure. Because what you don't really probably look at, or probably have never been explained to you before, is that these nitrogenous bases, so as I said, right, sugar, phosphate, sugar, but off the other end of the sugar is the nitrogenous base, right? And we have 4 different ones for DNA. You have an adenine, a thymine, a cytosine, and a guanine. And if you look at the chemical structure of these, do you notice that adenine has 2 rings and thymine has 1, cytosine has 1, and guanine has 2. So this is a little bit larger molecule, these ones with the 2 rings, right? And the ones with just 1 are, of course, a little bit smaller in comparison. This is partly why A's bind with T's and C's with G's. Because if you look at when it's double-stranded across one of what I sometimes refer to as the steps, right? Because you could almost imagine this as a spiral staircase, right? Where the sugar phosphate bonds create what we call the backbone, right? Which is like the railing or banister going up the spiral staircase. Can you guys envision that? Okay, and then each one of these nitrogenous bases binding with its complementary is like the steps of that staircase. So do you see, when you look, you have G, which is 2 rings, binding with C, which is 1 ring. So you always have 2 with 1, so you could almost say that it's 3 rings across. So imagine the width of that step on that spiral staircase is, say, 3 feet. The same is true when you look at the AT binding, right? It's T with A. 2 rings of A and 1 ring of T. So you have this nice, beautifully, architecturally, and physically sound spiral staircase. That's what DNA is, right? This beautiful spiral staircase. Now, what if we tried to put the 2 single rings together? What if we did thymine with cytosine? How many feet across would that, what would be the width of that step? It would only be 2, right? And if we put the 2 2-ring ones together, right, adenine and guanine, we'd end up with a step that's 4 feet wide, right? If you told a carpenter, I want a spiral staircase where some of the steps are 2 feet wide and some are 4 feet wide, what would he tell you? Go hire somebody else, right? Can't be done. It literally physically cannot be done. It would not be structurally sound. So does DNA need to be structurally sound? Absolutely, otherwise it would fall apart, right? So that's pretty much why they bind the way they do. And then also the interactions between each one of them. They actually bind by what type of binding across the 2 strands, the 2 nitrogenous bases. What do we call that bond in between them? It's a hydrogen bond. And if you look, that's denoted by the dotted lines here, right? Notice between G and C, there are 3 pairs of hydrogen bonds. Between A and T, there are only 2. So sometimes you'll hear people talk about GC rich or AT rich regions. So region in the DNA where there's a lot of G's and C's as opposed to A's and T's. Which do you think is held tighter? G's and C's or A's and T's? G's and C's because what? More binding, right? They've got 3. So you'll notice, what is it called? The ta-ta box, right? Where usually they start to separate the 2 strands to replicate it. It's called that because it's mostly what bonds? A's and T's. Less hydrogen bonds, easier to pull apart. So that's why, right, chemically they combine with each other, and physically it all ends up being even, right? Beautiful 3 feet across spiral staircase. So it's like trying to, build Legos and the piece just doesn't fit, right? When you're trying to build a wall or something, right? You gotta get a different size piece so it'll all be uniform. So sometimes when this DNA is being copied, it'll accidentally put the wrong one there. And so if you switch one class for the same class, it's not a big deal. Like, right, if you switch a C for a T, you still got 3 feet across, right? Those are called transitions, right? When they switch one class of nitrogenous base for the other. That class, which I don't make you guys know for this class, but like if you're going to go on in genetics and stuff, these guys with the single ring are in a class known as pyrimidines. The ones that have 2 rings are known as purines. And I learned a saying a while back that helps me remember that, is angels are pure, right? So AG for adenine and guanine, right, are pure, like purines, right? See, play on words. And then how I remember they have 2 rings is that wings, W-I-N-G, right? Wings rhymes with rings, right? These circular structures that chemicals can form. And so purines, right, which are related to angels in my little story, and play on words, have 2 rings, 2 wings, so therefore purines have 2 rings. It's like a whole bunch of rhyming there though, huh? Don't need to know it for this class, right? But if you're going to continue on in things like genetics or biochemistry, it's helpful to know which ones, right, have how many rings in their structure. Yeah, adenine and guanine are purines, and they have 2 rings. And then pyrimidines have a single ring. And also in that class is uracil, right? Uracil is a single ring, but that's only found in RNA. So if you do a transition, no big deal as far as the structure of DNA. If you do a transversion, which means they switch maybe a C out for an A, so now you have 4 rings across, that'll kind of cause a buckle in the DNA. And that's kind of really a red flag for the enzymes associated with the DNA. They're going to be like, oh, something's not right. Right? I might need to fix this, because it's the structure of the DNA itself will be distorted if you switch between the different classes of nucleotides, a pyrimidine for a purine. So that's why I explained that one to you guys, so that when you read that in the book, you understand what they mean. So I don't think they actually do. They might do a good job explaining it when they explain the structure. I honestly skimmed over that part in the book this week because I know it by heart. I feel like reading it again. All right. You guys feel comfortable with that? So it's important, right? There's a reason why A's bind with T's and G's with C's. And then the other little story I tell myself is A, T, and T, right? They're still around. A's go with T's, A, T, and T, right? And C's and G's kind of look like each other. They're both curved, so it kind of makes sense that they go together. That's how I remember C's with G's and A's with T's. Whatever works for you, right? Whatever saying you learned when you were in biology or didn't learn, make yourself up something so you remember, because it's important. So although the structure itself, right, A's with T's, double-stranded, right, sugar phosphate backbone, is the same in all the 3 domains. Which, what are the 3 domains of life? What's the A one? Archaea, B, bacteria, and E, eukarya, which we're in, right? That domain. So bacteria have a single circular chromosome, right? So long molecule attached to itself in a circle. Most, of course, there are exceptions to that, right? Most of them have a circular chromosome, a single piece of DNA in a circle. Of course, it's a really long piece of DNA, right? But it's still just one for most bacteria. Same is true for most archaea. The difference is, as we said, they do wrap theirs around histone proteins, Not the same ones in eukaryotes, but very similar. Something you don't see in bacteria. In eukarya, this is where we get different too, right? We wrap around histones, but our chromosomes aren't circular, right? They're linear, and there's multiple linear chromosomes, right? We don't just have one chromosome. For people, how many chromosomes do we have? 26 pairs, right? Because we actually only have 23, but they come in pairs, right? Because we're what are called diploid. And that's important too, because bacteria and archaea are haploid. They only have one copy of most of their genes. So this is just in summary, and then of course there's some notable differences out here, right? So Borrelia burgdorferi, anyone know what disease that one causes? I don't remember if I put that one on the test. Borrelia burgdorferi. It actually has linear chromosomes even though it's a bacteria. It's a spirochete, causes one of our selected diseases that we're talking about in this class. Yep, Lyme disease. Lyme disease. And plasmids, remember we mentioned those extra pieces of DNA? Very common. So in addition to their chromosomes, circular pieces of DNA. Extra stuff that they don't have to have, but they can have. It's very common for bacteria and archaea to have them. It's relatively rare in eukaryotes, but they have been found in fungi and in some plants. Particularly for the fungi, they've been found in yeast, single-celled fungi. So notice it says very common but rare. And then the histones are only in archaea and eukarya, not in bacteria. Bacteria and archaea, single strand of circular DNA. We're linear for us, and then we have the two cycles, right? Where we're 2N or diploid, and then when we make our sex cells, they're haploid. Then they combine to be diploid again. Where for these guys, right, they just make a copy and give it to their cells. They get, their daughter cells get everything that they have, right, for the most part. They just make a copy and split. We call that binary fission. Where for us, right, for eukaryotic cells, what two replication of cell methods do we have? They both start with M. So when our somatic cells divide and they just make exact copies for the next daughter cell, that's mitosis, right? And when we make our sex cells, meiosis, right? Where we actually divide the information in half, right? And in that case, each sex cell is different depending on how you divide up the pairs. So of course there's always exceptions. So this guy was in the chapter opener, right? And he forms typically these groups of 4 cells. What do we call that? What arrangement is that? Tetrads, right? He forms tetrads. But within each of these cells, although bacteria usually only copy their DNA when they're getting ready to divide. But instead, this guy will keep anywhere between 4 and 10 copies of his genome inside a single cell. So instead of having the usual 1 copy in 1 cell for bacteria, which I don't remember if this guy's classified as a bacteria or an archaea, but he's definitely a single-celled eukaryote. He has 4 to 10 copies.
So each of those copies become a new one?
Yeah, they're all the same. Right? So it's like 10 copies of the same quiz. And they stack up and form this like donut shape. What's interesting about this is that the environment that we'll soon learn, things like UV radiation and radiation in general, different chemical mutagens out there, this guy can survive radiation damage up to 100 times more than any other cell like it. Yeah. So why is radiation bad for us? Why do you not want to go get like an x-ray, you know, every day? You can die, but why do you die? It's damaging something, isn't it? What is it damaging that you need to not be damaged? Yeah, it damages the DNA. That's how it kills the cells. That's what ionizing radiation does. It literally will cut the DNA up into pieces. Not good. No, they're throwing electrons at you. And they just, like little knives, cut it up. Yeah, this is, you know, an example to help you understand the fact that radiation destroys DNA.
So there's no way to improve that once the radiation is damaged the DNA, right?
Yeah, so we have some repair mechanisms, which is why you don't die, but that's also why like technicians leave the room every time you get x-rayed, right? Because otherwise they'd be irradiated numerous times every day and they would be dead in a year. And it's also why if you're not getting irradiated in this area, they cover you up with a lead vest so the radiation doesn't penetrate through to what? What are they covering up in this area of your body? Your reproductive organs. The reproductive organs. Because again, you don't want to damage the DNA of your sperm and your eggs that you're going to, you know, make babies with. Yeah. Yeah. Damage to DNA. That's a lead vest to protect the rest of your body from the radiation. So they're only irradiating what they need to irradiate. Probably.
I was wondering, what happens like if you go to the dentist office and you have a lot of x-rays, you know, dental x-rays, or you break bones, or
yeah.
you do that often, does that cause damage?
It does, and that's why you don't want to do it too often. And they keep track of it. Yeah, but they're going to do that once. They're not going to turn around and do it the next day and the next day and the next day, right? They're going to do it once. And that's also when you're pregnant. This is why they're always asking all the time, are you pregnant, right? Because they don't want to irradiate a baby. Yeah. Yeah. Because it does damage. It does do damage. So now you know. So why does this guy have like 4 to 10 copies? It's like spare copies. It's protection, right? So if you irradiate him, he's like, no problem. That copy's no good. I'll use this other copy. I got a spare. I'm all set. Right? So this is an adaptation, right, of surviving potential damage to his DNA. He's like, I'm just going to keep extra copies just in case. Right? Does that make sense to you guys? So how many of you guys, clearly by the discussion, knew that x-rays damage your DNA before I told you? One person. So now you know, right, why we do these things. So when we replicate it, and again, we're not going to get into all the particulars in this class, you know, what enzymes untwisted and what ones bind, and oh my God, no. The important thing is right, enzymes attach. We separate the strands into 2 different sides, right? And we use each of the parent strands as a template for the new, right? And that's what they're trying to show you in this colored picture. So here's the original. So this is said to be semi-conserved, right? Because you have one of the parent strains associated with the new strain, right? And then that's passed on. And then for the next generation, the very first parent strain, of course, isn't there eventually. It's all new stuff. But using one side as a template for the other, they're having this complementary base pairing, right, makes it really easy to copy this information. So this is an important question, though. How does the cell know which strand is old and which strand is new? So let's this is just like photocopying a paper, right? If I go into the office and I put a paper on the copy machine and I spit out a copy, I walk away. Is there any way I can tell the difference between the copy and the original? Sometimes, right? We kind of, it looks a little different, right? When you copy something than the original, especially if you wrote in colored ink and it's not a color copier, you'll definitely know the black and white. Oh, that's the copy. But they have to have some way to label it, right, in your cell, old versus new. So what they do is they methylate, they add carbon and hydrogen groups to the old DNA. Right? As the DNA matures, they add methyl groups. So they basically label it old, 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 old. I don't understand the question. No, it's not. It's not old and we're not getting rid of it, right? We save it. It's the template for the next, right? So all this DNA in this parent cell has been methylated. So when the two strands separate, they know, like here we have it color-coded, but they chemically code it, right? If you were to look down to the molecular structure, this blue strand is methylated. And this blue strand is methylated. Where the new strand is not methylated, because it just made it. So enzymes, when they go through and they're checking the code to make sure that it's correct, they know that the template strand, the old strand, the methylated strand is what it's supposed to be. And if the new strand doesn't match, then they fix that. You see? Still with me? The problem is if they don't fix it, right? Then it gets methylated and we pass it on to the next generation. And they think, oh, that's the way it's supposed to be. That's how mutation happens, right? Okay. So there are many enzymes, right, across the domains that actually replicate, actually copy that sequence. By no means are we memorizing these, right? I don't even know half of them. But in general, right, they're referred to as DNA polymerases for the most part, right? So they polymerize, they add the nucleotides together of DNA. The important thing about some of these enzymes, especially DNA polymerase, whether it's DNA polymerase 3 or 1, or, the replication one is DNA polymerase 3 in bacteria. It's bDNA polymerase and polymerase DNA. They all say DNA polymerase somewhere in their name, right? But they're all slightly different from each other in the different domains. They all do the same thing, though. They copy DNA. And they do one other really important feature that I talked about last time. Nope, they're just copying, right? But when you copy stuff, do you always do it right? When you copy stuff down? Sometimes you make mistakes, right? Do you catch sometimes when you make mistakes? Yeah, sometimes right away, and you fix it right away. So guess what? The one that makes DNA can also fix its own mistakes if it catches it. It doesn't always. It's not perfect, just like us. But it's pretty darn good. No, that exact same enzyme that makes the DNA polymerase, can also fix its own mistakes. So it not only makes DNA, but it also fixes, it just does what we refer to as proofreads its own work. And this is why, one of the reasons why, other than other enzymes that we'll talk about later in repair mechanisms, why we're not all, crazy mutated X-Men. Because we try not to have mutation happen. But it does happen, and that's why organisms change. And with selective pressure of the environment, you get new forms of organisms, right? And the really scary thing, and especially why we talk about it in this class, is as it relates to the DNA mutating in bacteria and then become resistant to antibiotics. So remember I told you guys I was going to find that link. I did find it, and I am going to post it for you guys for this class. I posted it, of course, in my genetics class yesterday. I'm going to copy it over for you guys. It's the giant petri dish, right?